The 1860s were a time of rapid change, a period of unprecedented progress and innovation that touched every facet of human life. Steam engines roared, powering factories and transforming industries, and scientific revolutions were brewing in laboratories across the globe. New discoveries were constantly reshaping our understanding of the world. Scientists, peering through newly improved microscopes, could see cells, the fundamental building blocks of life, with greater clarity than ever before. But a fundamental question remained. How are traits passed from parent to child? What is the mechanism of heredity? Theories abounded, speculative ideas filled the scientific literature. Was it a blending of blood, a literal mixing of parental essences, or was it something else entirely, something more complex and nuanced? Careful observations, however, didn't quite fit the old ideas. The prevailing theories struggled to explain the complexities of inheritance. Traits skip generations, defying simple blending, and children often resemble distant ancestors, hinting at a more intricate process at play. Darwin's groundbreaking theory of evolution by natural selection made heredity a central pillar of understanding the natural world, but the underlying mechanism of how traits were inherited remained a profound mystery, a puzzle that captivated the minds of scientists. Most scientists at the time believed that proteins, with their immense complexity and diverse structures, must be the carriers of heredity, the molecules responsible for passing traits from one generation to the next. Proteins seemed to fit the bill perfectly. They were complex, incredibly abundant in cells, and absolutely essential to all known forms of life. Few scientists at the time even imagined that another, less understood substance could possibly be responsible for this fundamental process. Across Europe, and indeed around the world, scientists attacked the problem of heredity from all sides, employing a variety of experimental approaches. Some, like Gregor Mendel in his quiet monastery garden, meticulously bred pea plants, patiently uncovering the fundamental laws of inheritance through careful observation and statistical analysis. Others, armed with increasingly powerful microscopes, watch cells divide and multiply, searching for the elusive hereditary material within the intricate structures of the cell, hoping to catch a glimpse of the secret of life. It was a time of intense curiosity and relentless investigation, a period of fervent pursuit of knowledge, with everyone searching for the invisible architect of life, the hidden blueprint that dictated the form and function of every living thing. The stage was set, the players were in place, and the world was poised for a discovery that would fundamentally change our understanding of biology and life itself. Into this world stepped Friedrich Miescher, a quiet, meticulous Swiss scientist. Hearing loss pushed him from medicine to physiological chemistry, where he could focus on the cell's inner workings. In 1868, he joined Felix Hoppe Seyler's lab in Tübingen, Germany, a hub of cutting-edge research. Miescher was assigned to study the mysterious cell nucleus, a daunting task no one had yet mastered. His patience and precision made him perfect for the job. He wasn't seeking fame, just truth, determined to break the cell down to its fundamental parts. Miescher's relentless pursuit would soon lead to a discovery beyond his wildest expectations. In the solitude of the lab, he found his calling. He would become the first to glimpse the true substance of heredity. The quiet scientist was about to make history. To truly delve into the mysteries held within, Friedrich Miescher needed to study nuclei. But not just any nuclei. He needed cells with exceptionally large and prominent nuclei to facilitate his investigations. This quest for suitable cells would lead him down an unexpected path. In a rather unconventional turn, he found an abundant source of such cells in pus-soaked bandages collected from a local clinic. These discarded medical dressings, teeming with biological material, held the key to his initial experiments. The pus was rich with white blood cells, also known as leukocytes. These cells, crucial for the body's immune response, were abundant in the infected wounds and possessed the large nuclei Miescher sought, making them perfect for his meticulous experiments. With his source secured, he embarked on a painstaking process. He washed the cells to remove debris, carefully separated them from the surrounding material, and then gently broke open the cell membranes to access their precious contents. His primary goal was meticulously isolating the nuclei, the command centers of these cells. This required delicate techniques and unwavering patience as he sought to purify the nuclei from all other cellular components. During the course of one such experiment, as he was carefully analyzing the nuclear extracts, he noticed a peculiar, strange gray substance that precipitated out of the solution. This substance exhibited unusual properties. It didn't behave like any known protein, the primary focus of biochemical investigations at the time. Its characteristics defied expectations. Further investigation revealed that it was insoluble in acidic solutions in alcohol, common solvents used in laboratory procedures. This insolubility set it apart from many other biological compounds. However, in stark contrast, the substance readily dissolved in alkaline solutions, indicating a unique chemical composition and behavior. This peculiar solubility profile piqued Miescher's curiosity even further. Intrigued by these unusual characteristics, Miescher made a pivotal decision. He shifted his research focus to this mysterious material, determined to unravel its secrets and understand its nature. Through a series of meticulous chemical tests, he discovered that the substance was remarkably rich in phosphorus, an element not typically found in high concentrations in proteins. This high phosphorus content distinguished it from the known biological macromolecules of the time proteins, fats, and carbohydrates, suggesting it was something entirely different. It was at this moment that Miescher had a profound realization. He had stumbled upon a completely new class of biological molecule, unlike anything previously described in scientific literature. He christened this novel substance nuclein, a name derived from its origin within the cell's nucleus. While he had successfully isolated and characterized this new molecule, he had no idea what its function might be within the cell or its broader significance in the realm of biology. Despite the mystery surrounding its purpose, Miescher recognized that nuclein was fundamentally new, a distinct entity with unique properties that warranted further investigation. The fact that this novel substance originated from the cell's command center, the nucleus, hinted at its potential importance in cellular processes and the regulation of life itself. In his meticulous experiments, Friedrich Miescher had unknowingly uncovered a hidden player, a crucial component in the intricate story of life, setting the stage for future discoveries. Little did he know at the time, but his discovery would prove to be of immense importance, laying the foundation for our understanding of genetics and heredity. The secret of heredity, the mechanism by which traits are passed from one generation to the next, was now closer than ever to being revealed, thanks to Miescher's groundbreaking work. Nuclein was a puzzle, unique, acidic, and packed with phosphorus. Its chemical profile set it apart from all known molecules. Why was it locked away in the nucleus, the cell's control center? Miescher wondered if it was just a phosphorus store, not knowing it was the backbone of a genetic code. 19th century science couldn't decipher its structure or function. It was like finding a book in an unknown language, its story unreadable. Scientists saw its importance by location, but not its purpose. Nuclein was a mystery waiting for future generations to solve. The molecule's true significance was still hidden. Miescher's discovery didn't spark immediate excitement. His mentor, Hoppe Seiler, insisted on repeating the experiments before publishing, delaying recognition for 2 years. Other students confirmed the findings, and in 1871, Miescher's paper finally appeared, but its impact was muted. The scientific world was fixated on proteins, dismissing nuclein as too simple to carry heredity. Early analyses suggested it was repetitive and unremarkable. Miescher, shy and reserved, didn't promote his work. He continued studying nuclein in salmon sperm, refining his analysis, but the world paid little attention. His discovery languished in obscurity, overshadowed by prevailing theories. The molecule of heredity was hiding in plain sight. It would take decades for its importance to be recognized. Miescher's quiet breakthrough waited for its moment. While Miescher worked in Germany, Gregor Mendel was uncovering the laws of inheritance in a monastery garden. Mendel's experiments with pea plants revealed that traits are passed as discrete units, what he called factors. He discovered the mathematical rules of heredity, showing traits are dominant or recessive and inherited independently. Mendel described the how of inheritance, but not the what. Miescher, meanwhile, had found the what, but not the rules. The two men, unaware of each other, had solved different halves of the same mystery. Mendel's work, like Miescher's, was ignored for decades, too abstract for most biologists. The world had both the rules and the molecule, but hadn't connected them. Their discoveries would only be united much later. The foundation for modern genetics was quietly laid. Friedrich Miescher's groundbreaking discovery of nuclein, a substance distinct from protein, marked a pivotal moment in the history of biology. It was the very first step toward truly understanding life's molecular basis, opening up avenues of research that would eventually lead to the unraveling of the genetic code. This discovery was more than just an observation, it was a paradigm shift. He proved, through meticulous experimentation, that the cell nucleus held more than just proteins, the molecules then believed to be the sole carriers of hereditary information. He found a new, fundamental molecule, a previously unknown component of the cell with properties unlike anything seen before. This was a scientific revelation. Later, scientists like Albrecht Kossel built upon his foundational work, expanding our understanding of this mysterious substance. Kossel played a crucial role in identifying the nitrogenous bases, adenine, guanine, cytosine, and thymine, the building blocks that make up DNA's intricate code. Miescher found the book he identified the key ingredient. Kossel began to read the letters, deciphering the molecular alphabet that would eventually reveal the secrets of heredity. His work challenged the prevailing protein-centric view of heredity, a view that had dominated scientific thought for decades. Kossel's research was instrumental in keeping alive the fundamental question: what if heredity's secret, the very blueprint of life, wasn't solely contained within proteins? Nuclein's unique and consistent location within the cell nucleus and sperm cells, the very vehicles of inheritance, strongly hinted at its profound importance in the transmission of genetic information. Miescher's patient and meticulous work anchored an alternative idea, a seed of scientific thought that would eventually blossom and triumph over established dogma. Every great journey of discovery, every leap forward in scientific understanding begins with a single crucial step. Miescher's discovery of nuclein was that essential starting point, the foundation upon which the entire edifice of modern genetics would be built. He gave the molecule a name, providing it with an identity within the scientific lexicon. He pinpointed a location, placing it firmly within the cell's control center, and he defined a chemical identity, classifying it as a unique and distinct substance. And so, the foundation of modern genetics, the very bedrock of our understanding of life itself, was painstakingly set in a cold castle lab. Miescher's thread was picked up by others, slowly revealing the secrets of heredity. Early 20th century work linked genes to chromosomes made of protein and nucleic acid. For decades, proteins were favored as the genetic material. In 1944, Avery, McLeod, and McCarty showed DNA, not protein, carried genetic information. DNA finally took center stage. The race to uncover its structure began. Franklin's X-ray images revealed its helical shape. Watson and Crick, using Franklin's data, built the double helix model in 1953. The structure explained how DNA stores and copies information. Miescher's discovery, Mendel's laws, and decades of research came together. The secret of life was revealed. The legacy of nuclein had finally unfolded. Today, it's hard to overstate the impact of Miescher's discovery. He didn't know he'd found the blueprint of life, but his careful work opened a new world. Modern biology, medicine, forensics, and agriculture all trace back to that first isolation of nuclein. We can now read the story of our species and all life thanks to his work. Miescher's story is a lesson in scientific progress, slow, often overlooked, but world-changing. Revolutionary ideas can take decades to be understood. Sometimes, the greatest discoveries come from patient observation, not grand theories. Miescher wasn't seeking the secret of life, but he found it. The first page of the Book of Life was his gift to us. The story he started has transformed our world and our understanding of ourselves.